DNA contains two strands of building blocks called nucleotides, arranged like a spiral staircase. Each nucleotide includes three parts: a phosphate group, a sugar molecule, and one of four bases: adenine, guanine, cytosine, or thymine. The sugar phosphate bonds form the double backbone of the molecule, the handrails of the staircase. But we find the genetic key to DNA in the steps of this stairway, the nitrogen-containing bases. These bases link up using hydrogen bonds in a very specific way. Adenine will bond only with thymine, A to T. Cytosine only bonds with guanine, C to G. While these basic pairings never change, the order of the pairs along each strand varies greatly from one species to the next. In this elegant design, we see how nature stores the instructions to build all living things.
Did you know? Each human cell contains millions of bits of DNA. Each bit is a piece of information which controls how the cell works and ultimately what we look like. DNA contains the 4 letters used in a code, the universal language of life itself.
To make perfect copies of our DNA, an enzyme first unwinds the two strands of the molecule by separating its base pairs. Another enzyme then attaches free nucleotides to the exposed sites. The existing strand determines the composition of the new strand. According to the base pairing rules, adenine bonds with thymine and guanine with cytosine. In long strands of DNA, replication occurs in many regions simultaneously, forming the sister chromatids required for cell division.
Inside living cells, the actual process of making proteins involves several different steps because the instructions for making proteins are stored in the nucleus, but the proteins themselves are made or synthesized in the cytoplasm. This means cells must have a way to move information out of the nucleus and into the cytoplasm. As it turns out, cells use a special molecule for this task, called messenger RNA. RNA, or ribonucleic acid, is very similar to DNA, but RNA has only a single strand. It contains the sugar ribose instead of deoxyribose, and a base called uracil replaces the thymine found in DNA. So the RNA alphabet is A, U, C, G, not the A, T, C, G used by DNA.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
A mutation is something that has gone wrong with DNA. The genetic code is a code of words, each of 3 letters. My favorite example is, the fat cat saw the red dog run out. If you take out the FA of fat, the rest of what you have in that sentence is now nonsense.
A point mutation is a change in the nucleotide sequence of a gene. This can result from an error during replication that is not corrected by the usual repair mechanisms. Under normal circumstances, adenines and thymines should bind to one another, as should cytosines and guanines. In a base substitution mutation, a non-complementary nucleotide is incorporated into the new strand. The resulting altered triplet may code for a different amino acid, or even a stop codon. Here we see 4 codons and their corresponding amino acids. A change within the second triplet results in a change in the amino acid. None of the other amino acids are affected. Insertions and deletions result in frame shift mutations. The insertion of an extra nucleotide not only affects the triplet it is part of, but all subsequent triplets, thus drastically altering the protein product. The result is similar if a nucleotide is deleted.
happy because he has diabetes and needs insulin to stay healthy and happy. Today we are going to make insulin through a genetically modified organism. Your DNA gives you the characteristics you have, like your eye color. Your eye color and your other characteristics are caused by the genes in your DNA. Here is the gene for eye color. Your DNA also determines your hair color. And here's the gene for hair color in your DNA. An organism is genetically modified when its genes have been changed. There is also a gene in your DNA that makes insulin. Insulin is what PRAME needs. So, to get this gene, we use restriction enzymes to cut it out. Restriction enzymes are like scissors. They cut your DNA at certain sections. Here is the insulin gene. This is a cell of a bacteria, another type of organism. In bacteria, some of the DNA is in circles. These are called plasmids, and we can use them to insert a gene into the bacteria. In order to insert the insulin gene into the plasmid, we need to cut the plasmids with restriction enzymes, or the scissors. Then we need to insert the insulin gene into the plasmid. Another enzyme called ligase helps us to do this. Ligase is like a glue stick and glues the end of the insulin gene to the plasmid. Then we can put the plasmid with the insulin gene back into the bacteria. The bacteria is now a genetically modified organism because it's a living thing and we have modified or changed its genes. The bacteria duplicates again and again to make many bacteria with the insulin gene. The bacteria have a human insulin gene, so they make human insulin. The scientists can take this insulin and give it to people with diabetes, like PRAME, so that they can be healthy and happy.